Again, the next topic is neurotransmission. Neurotransmission means transfer of action potential from one neuron to another neuron, or from one cell to another cell. That will be better and comprehensive also. So, how this message we have covered that how the signals are sent through the axons. But what happens when the signal reaches, or when action potential reaches to its terminal, and how it is transferred to the next cell or next receptor site? Uh, First, here we have an image which we have to understand. Then we will be able to know how this happens, this process, about this process. This is a synaptic terminal. Synaptic knob. Knob means everything which is somehow swollen. Okay, so this is a synaptic knob, and in this synaptic knob there will be action potential, let's say AP, and there are some synaptic vesicles which are in this synaptic terminal, like these are indicated by this arrow. So these synaptic vesicles are actually blisters which have some liquid chemicals known as neurotransmitters. These synaptic vesicles are some blisters, something like blisters, in the synaptic terminal, and they contain some liquid chemical molecules named as neurotransmitters. The gap between one neurotransmitter, between synaptic terminal and the receptor site of another neuron or another cell, is known as synapse. This particular space will be known as synapse, and this is your entire receptor site. And this receptor site, there are some spaces which are made to fit the neurotransmitters. Neurotransmitters are released, and some of them get fit into this receptor site. There are something you can understand it like there are some type of holes, some specialized holes of a special size, in which only a special kind of neurotransmitter will be fitted. This will be called, this synaptic terminal will be known as presynaptic neuron, and this receptor site will be known as postsynaptic or receiving neuron, and sodium ion, as we have discussed earlier. Now what happens during the what happens when action potential reaches to the axon terminal? When it reaches to axon terminal, from these synaptic vesicles, some neurotransmitters are released. They are of different kinds, but for understanding purpose, we will use it for only one kind, until and unless it is required to elaborate that they may be of different kind. Okay, so different kind of so neurotransmitters will be released here, and this synapse is filled with some with a liquid sap. They will start, these neurotransmitters will start floating in this synapse, and after some time or less than some milliseconds, they will get in touch with the surface of receptor sites, and when the some of them some of the neurotransmitters will then fit into the holes of these receptor sites. As soon as these receptor sites are filled with these neurotransmitters, sodium ions will now move rapidly into the axon of the next cell so that action potential can be transferred. Remember, I am repeating again, as soon as these receptor sites are filled with neurotransmitters, sodium ions which are present outside the axon, they are positive in nature, they rush into the axon of the receptor site, and hence action potential continues to be delivered to another cell. So what happens with this neuron? Sorry, what happens with these neurotransmitters which are already released and are now resting with these receptor sites? There is a process named as reuptake. It may be also called as selective reuptake. The process of reuptake works as a suction pump. Like this synapse will suck some of the neurotransmitter into it again, into its synaptic vesicle again. But some of them are not reuptaken, and they are broken down by some of specialized enzymes. Hence, the process is complete. This reuptake will start immediately when the neurotransmitters are fitted into the receptor site. As soon as the action potential has been transferred to the next cell, the reuptake process will be started. It will not take into the seconds, thousands of milliseconds, within less than 1 upon 10 to the power minus 100 or 100. This process will be started, means it will take some of the milliseconds only to initiate the process of reuptake. Okay, so this has happened how remember this thing, that this receptor site need not to be only next neuron. Like this was a neuron, so should this receptor site must also be a neuron? No, this may be a neuron but may not be also. There are some muscles also who have some receptor sites for their contraction and for their relaxation, and like heart. So this is not essential that the next neuron or the next cell will be a neuron, because receptor sites are present in nearly every type of organ we have. The question is, we have talked about these neurotransmitters. What are these neurotransmitters? They are let's discuss about it. They are actually the chemicals. They are molecules of special chemicals which are used to transfer the signals from one neuron to another cell or one cell to another cell in an specialized way. What is the difference and why I said a specialized way? Let's make it clear. The first neurotransmitter which was discovered was acetylcholine. Remember, this name is very important. This may come in our MCQs also. It is found at the synapses between the neurons and muscle synapses. You know, And acetylcholine stimulates the skeletal muscle to contract but actually slows contraction in the heart muscle. When acetylcholine, a neurotransmitter, will be released from the synaptic terminal or synaptic vesicle of a neuron, it will stimulate the skeletal muscles to contract. Means it will act as an excitatory neurotransmitter. But when talking about heart muscles, it will reduce the contraction rate of the contraction of the heart muscle. So in the heart muscle, it will act as inhibitory. It will act as inhibitory in your heart muscles. So it has both the properties. It will either enhance the effect of the signal, or it may also inhibit the effect of any action potential. When talking about the enhancing or inhibiting any effect, it doesn't mean that neurotransmitter itself is excitatory or inhibitory. Neurotransmitters are just a chemical elements or compounds. They themselves are neither excitatory nor inhibitory. They, but they have the ability to change the signal strength. That is, if signal strength is changed, it means the transmitter is has enhancing power, and it will create excitatory effect. And when it is reducing the impact of all the strength of the action potential, it means it is working as an inhibitory neurotransmitter. Very simple definition. What happens if acetylcholine reactors, actually it is shown by ACH, it is its short form. And if acetylcholine receptor sites on the muscle cells are blocked in some way, then the acetylcholine can't get to the site and the muscles will incapable of contracting. Means you cannot contract your muscles if acetylcholine has been blocked by the by any way, or they may be paralyzed. In other words, this is exactly what happens when curare, a drug used by American Indians on their blow darts, get into the nervous system. Curare molecules are just similar enough to fit into the receptor site without actually stimulating the cell, making curare on an antagonist, a chemical substance that blocks or reduces the effect of neurotransmitter. Now here two other words. I have told you about uh, excitatory synapse, and inhibitory synapse. Now there are antagonist and agonist. Remember, neurotransmitter will create an effect on the on the strength of action potential or the effect of action potential. Now what these antagonists will do? Antagonists are external substances which are not belonging to our natural body part. It may be a drug, or agonist will also be something like a drug or anything else which is not related to body. Now this antagonist will work opposite to the effect of the neurotransmitter. If acetylcholine is creating some excitatory effect, then if we use curare as an antagonist to the acetylcholine, then the effect of acetylcholine will become inhibitory, or the acetylcholine will be blocked and only curare will work. That's why it will inhibit the contraction of the muscles. And another one is antagonist, which will work as per the effect of neurotransmitter. Means if neurotransmitter is excitatory, then it will further excite, enhance the effect of that action potential. And if neurotransmitter is inhibitory, then this agonist will further reduce the effect of that action potential. This is how they work. They both are related to drugs, and antagonist will work opposite to the effect of the neurotransmitter, whereas agonist will always work in the direction of neurotransmitters. Have some knowledge also about what is the working of first we knew that acetylcholine can work as an excitatory or inhibitory neurotransmitter. But what are its effects? Let's discuss it. Uh, what happens if the neurons release too much acetylcholine? If less acetylcholine is released, then what happens? It is we cannot contract our muscles. That happens. And what if this release is too much? Amount of acetylcholine has become great. The bite of black widow spider does that. Its venom stimulates the release of excessive amount of acetylcholine and causes convulsions and possible death. Black widow spider venom is agonist. Curare will act as antagonist to the acetylcholine, whereas the venom of black widow spider will act as agonist to the acetylcholine. When any black venom black widow spider bites someone, it will cause the muscles or the neurons to release too much of acetylcholine, and that stimulates the muscles, and due to so much contraction in a very limited time, death may happen. Acetylcholine also plays a role in memory, arousal, attention. For example, there is a region in our brain known as hippocampus which is associated with our memory and especially associated with the formation of new memories. And if low level of acetylcholine is found in the hippocampus, it will cause a disease named as Alzheimer's disease, which is the most common type of dementia. So if someone is feeling with Alzheimer's disease, he may be given an agonist. Next one is dopamine, which is also a neurotransmitter. These are important neurotransmitters which we are studying. Otherwise, there are a list of hundreds of neurotransmitters found in our body. So we are not going to detail in those type of neurotransmitter. We are reading only about which one is important. Dopamine is a neurotransmitter found in the brain, and like some of the other neurotransmitters, it can have different effects depending on the exact location of its activity. For example, if too little, same thing is, it also can work as excitatory and inhibitory. That depends on the location of the dopamine where it is found. For example, if too little DA is released, dopamine is referred to as DA in its short form. आ रहा हूँ रुक जा 10 मिनट दे तो मैं पहुँचता हूँ. If too little DA is released in a certain area of the brain, result is Parkinson's disease. But this is a very serious disease due to which there are many celebrities who are suffering right now, but we are not going to into the detail. Next one is, if too much DA is released in other areas, the result is a cluster of symptoms that may be a part of schizophrenia. The release of too much dopamine may be a cause of schizophrenia or may enhance the effects of schizophrenia. Next one is serotonin. It is a neurotransmitter originating in lower part of the brain that can have either an excitatory or inhibitory effect depending upon the particular synapses being affected. It is associated with sleep, mood, anxiety, appetite. For example, low level of serotonin activity have been linked to depression. If serotonin is less, it may cause a depression, or when we get into the depression, release of serotonin becomes less. Although ACH was the first neurotransmitter found to have excitatory effect at the synapse, the nervous system's major excitatory neurotransmitter is glutamate. Glutamate is an excitatory neurotransmitter which is a major which forms a major part of our nervous system. Like ACH, glutamate plays an important role in learning and memory, and may also have a role in development of nervous system and in synaptic plasticity, the ability of the brain to change the connections among its neurons. However, an excess of glutamate results in overactivation, and neural damage may be associated with the cell death that occurs after the stroke or head injury, or in degenerative diseases like Alzheimer's disease and Huntington's disease. These are factual data which you have to remember. Another one neurotransmitter is gamma antibutyric acid. It is also known as GABA, whereas glutamate is the major neurotransmitter with an excitatory effect. GABA is the most common neurotransmitter producing inhibition in the brain. So it will create inhibition, means it will mimic the effects of action potential. GABA can help calm anxiety, or example by binding to the same receptor sites that are affected by the tranquilizing drugs and alcohol. In fact, the effect of alcohol is to enhance the effect of GABA. Alcohol will work as an agonist to the GABA, because GABA is also already an inhibitory neurotransmitter. So if we use alcohol, it will cause to release more GABA, and hence the effects of action potential will be reduced at each stage of transmission. Here are some table is given where some of the neurotransmitters with their functions are given. Remember two things: glutamate, acetylcholine, and GABA. GABA is one which has only inhibitory effect, and glutamate has only excitatory effect. Other one are both inhibitory as well as excitatory. Reuptake, I have covered it. Here something is given for the medication purpose of the how drugs are how drugs work, and it is related to the release of neurotransmitter. Actually, drugs that work, for example, let me have an example. It will make you clear about this entire topic. You have anxiety. This will cause less release of serotonin. Less serotonin. Right. Now, due to this less release of serotonin, you may go to depression. You have what is the medication, the cure of this type of depression? You have to cause your cells, to cause your neurons to release more serotonin, and here's the solution has been given. Here it is given. Some drugs yield agonistic or antagonistic effects by impacting the amount of neurotransmitter in synapse. They do so by interfering with the regular reuptake or enzymatic degradation process. Remember the neurotransmitter serotonin helps regulate and adjust people's mood, but in some people, normal process of adjustment is not working properly. Some of the drugs used to treat the depression are called SSRI, selective serotonin reuptake inhibitors. SSRI block the reuptake of serotonin, leaving more serotonin available in the synapse. Here, that this was telling that you have some anxiety, and due to this, less serotonin. Due to this, you may get into the depression. And to stop this, we have to cause this serotonin uh, release. How this happens? We can use SSRI. It is a drug which works as an agonist for the serotonin, and when we use this drug, this will cause the serotonin to stay into the receptor site rather than reuptake. Means it will inhibit the reuptake of serotonin, and hence if serotonin is available in the receptor site of a next cell, it will cause the action potential to be sustained, and hence for many days this if the reuptake is not happening, then depression can be avoided with this. So this was one example, and another was one was about GABA. Also we can use agonist for GABA, such we know that it acts as an inhibitor, and hence we can use an agonist for the GABA to reduce our anxiety or depression. That was it for this today's class, and we have learned how the signals are transferred from one neuron or one cell to another cell, and then we have studied deeply into the neurotransmitters. Now next one is about nervous system, which is fully on biology. Like you may have seen this graph, that is nervous system is divided into two parts: central nervous system, peripheral nervous system. Central is further divided into two parts: brain and spinal cord. Then we come peripheral nervous system is also divided into two parts: automatic and somatic nervous system. Whereas automatic nervous system is also divided into two parts: one is parasympathetic division and second is sympathetic division. And somatic nervous system is also divided into two parts: sensory system and motor system. We will study about it in our next class, so let's stop this class right now here.